Hi everyone, my name is Tina, and today I will be discussing my project, which is called The Impact of Cannabidiol on Trophoblast Metabolism and Function. So approximately 2 to 20% of women have reported using cannabis during pregnancy, with about 11.3% of women from Hamilton, Ontario, reporting using cannabis during pregnancy, indicating that cannabis use during pregnancy is becoming more prevalent in the recent years. Now, in our bodies, and in many different cell types and tissues, we have what is called the endocannabinoid system, or the ECS, and this is comprised of different enzymes and receptors and ligands, such as anandamide or AEA, THC, 2AG, which interact with cannabinoid receptors such as CB1, CB2, and many others. Now, this system is involved in many physiological processes. However, we will be focusing on how the system is integrated in reproductive tissues. So with that, we are looking into how the placenta forms during pregnancy, which the placenta is comprised of stem cells or trophoblast stem cells, which can either be undifferentiated or differentiated subtypes. Undifferentiated trophoblast cells are called cytotrophablasts, whereas as they differentiate, they become syncytiotrophoblasts. One important feature about stem cells, such as trophoblasts, is that they have mitochondria, and their mitochondria undergo different morphological changes, which confers functionality to the stem cells. So stem cells do require mitochondrial dynamics in order to undergo commitment, proliferation, and differentiation. So there are two different distinct phenotypes between these two subtypes of trophoblasts. So cytotrophoblasts have more elongated, well-defined lamellar cristae, and they have larger mitochondria and greater electron transport chain activity. Whereas as these proliferative, undifferentiated trophoblasts differentiate, they have a different mitochondrial phenotype, which is exhibited as smaller mitochondria. They have more vesicular cristae, they're more spherical and punctate, which just speaks to the mitochondrial function. Now, more specifically, mitochondrial dynamics can influence stem cell fate decisions. So mitochondrial dynamics are important when it comes to whether a stem cell decides to undergo self-renewal or whether it decides to undergo commitment, proliferation, and differentiation. There is such thing as a metabolic switch that occurs where more fragmented mitochondria switch to an elongated phenotype, which gives the stem cell cues to undergo differentiation. Now, this is a transient change that occurs. Once the stem cell has reached a state of quiescence or a state of self-renewal, it does adopt the fragmented mitochondrial phenotype. Now, our research question is looking into how CBD impacts mitochondrial dynamics and markers of differentiation in these trophoblast cells. Previous work from our lab has shown that THC, another component of cannabis, has led to decreased trophoblast fusion and increased mitochondrial fission. So there might actually be a link between the changes to the mitochondrial dynamics and the decreased trophoblast fusion. We don't, however, have any knowledge about what CBD is, how CBD affects trophoblasts, which is why we want to understand how CBD interacts with trophoblast mitochondria, how it could alter their metabolism, and consequently alter their function. So we worked with an in vitro trophoblast model, which to date the best model of trophoblast stem cells is the BUOB30 cell line. So they do satisfy the 4 defining criteria of trophoblasts, which are listed, and undergo fusion following CAMP stimulation. However, they are derived from a gestational choriocarcinoma, and they don't recapitulate the dynamic environment of the syncytium and the maternal-fetal interface. So those are some limitations to our cell line. So on the left side, we'll see the experimental design. So on day 0, we seed, and then we treat with 50 nanograms per mil of EGF to facilitate monolayer formation, and then we treat with 20 micromolar of CBD after 2 days, and then following 48 hours, we will harvest our cells for protein and PCR. So first and foremost, we conducted PCRs and Western blots for either mitochondrial dynamics markers or trophoblast differentiation markers, and found that CBD does alter markers of mitodynamics and trophoblast differentiation. On the left panel, we will see that marker of mitochondrial fusion is lowered in both cytotrophablasts and syncytiotrophoblasts, or there's a decreasing trend for syncytiotrophoblasts as well. And then for mitochondrial fission proteins, we actually do not see a difference between the cytotrophoblasts, but we do see a large decrease from the control to treated syncytiotrophoblasts. As well, on the upper panel, we'll see a marker of cytotrophoblast differentiation, which is lower, and on the bottom panel, we will see another marker of syncytiotrophoblas differentiation, which is also lower. Next, we look at how CBD decreases transcript levels of human chorionic gonadotropin subunits. Now, HCG is a hormone that's secreted by syncytiotrophoblasts, and this hormone secretion is directly proportional to the extent of fusion that the syncytiotrophoblasts have undergone, and it is also a marker of trophoblast function in general. So to see that CBD has suppressed the transcripts of this hormone is telling us that CBD can actually diminish trophoblast function. As well, we see that CBD does alter the endocannabinoid system enzymes and receptors that we have looked at. However, it does not seem to be changing the syncytiotrophoblasts. However, there is an increasing trend for the syncytiotrophoblasts. And finally, we also have started looking into how some of the receptors, the cannabinoid receptors, are affected by CBD treatment, and we have noticed that CBD treatment in both syncytios and cytotrophoblasts seems to be decreasing the levels of CB1 protein. So that is telling us that CBD might be just downregulating CB1 expression and altering any pathways that are downstream of CB1. So in conclusion, the CBD treatments appear to be altering mitochondrial dynamics markers and decreasing markers of differentiation in cytotrophoblasts and syncytiotrophoblasts. We also see that HCG transcripts are diminished, suggesting that CBD lowers trophoblast fusion and therefore their function. Overall, CBD acts either directly or indirectly on cannabinoid receptor 1 to downregulate its expression in both CTBs and STBs, while further downregulating ECS enzymes and receptors in the CTBs. Taken together, our findings do suggest that CBD potentially depresses the ECS in trophoblasts and compromises syncytiotrophoblaст function as a result. So our next steps are to assess the extent of differentiation by conducting immunofluorescent staining of CBD-treated trophoblasts and quantifying cell-to-cell fusion. This will give us a clearer picture of what is going on in trophoblasts, because markers of differentiation could be lower, but the qualitative assessment is still necessary. We also hope to quantify protein expression of these two differentiation markers and assess trophoblast function by performing ELISAs for key hormones secreted by syncytiotrophoblasts, including HCG, progesterone, and human placental lactogen. Finally, we want to conduct live cell imaging of CBD-treated cytotrophoblasts and syncytiotrophoblasts to observe and quantify mitochondrial dynamics in real time. Thank you everyone for listening to my presentation.